Support Microbe TV. It's time for our annual fundraising drive. We depend on your support to produce high-quality science videos and podcasts. And now is the time to help us. If our programs have appealed to your science interests, or if they have helped you in some way, please record a brief audio or video with your phone and tell us about it. Let us know how we empower your inner scientist. We'll use it for our fundraising efforts. Send it to incubator@microbe.tv. And for more information on how you can give us your support, go to microbe.tv/contribute. From Microbe TV, this is Immune, episode 87. I'm Vincent Racaniello, and you're listening to the podcast about the body's defenders against disease. Joining me today from Ithaca, New York, Cindy Leifer.
Welcome back. How are you doing, Vincent?
I'm okay. I'm back at the incubator, as you see.
I see that. Yay.
Do you have snow up there yet?
No. Okay, so it got a little bit colder, but it has been quite unseasonably warm.
Also joining us from Madison, New Jersey, Brianne Barker.
Hi, great to see you all. Also no snow, but that's less surprising. Also unseasonably warm.
I don't think we're going to have any snow down here, frankly.
Did we have much of any last year?
No.
Nothing.
Even in Ithaca, we really didn't.
It's crazy. Yeah. If you like these conversations about science and other things like weather, we'd love to have your support. This is giving season in November and December. Go to microbe.tv/contribute. In addition to the usual ways you can contribute, we have some things you can, like the NPR fundraisers. They give you a tie or a belt or something like that. We give you cooler things. So check that out, microbe.tv/contribute.
I was going to say I was in a cab at one point that was listening to one of those NPR things.
Yeah.
And that day they were giving away a chocolate babka. And according to the cab driver, the chocolate babka day was the day that they always made the most money. And it was by far the most popular thing.
How interesting. we don't give away any edible stuff. But,
we should make little chocolate viruses or something.
Yeah, that would be good.
Or macrophage cookies.
Also, if you're concerned about the recent nominee to head Health and Human Services, RFK Jr., and you should be concerned.
Yes.
I'm going to give you a link to a website where you can go, and it will send your letter to all the senators. So you can write something. And by the way, we just released a video on YouTube, a 5-minute video, which is a compilation of many things that Paul Offit has said on Beyond the Noise about why he would be inappropriate for this position. So safecommunitiescoalition.org/RFK. I have a page just for him. I was contacted, but this is a 501c3 nonprofit, safecommunitiescoalition.org. And the lady contacted me, said, I've been listening for years. It's the first time I'm contacting you. We need to spread the word about this website we just built. So safecommunitiescoalition.org/RFK.
And when you contact your senators and representatives, you might think that your one letter doesn't make a difference. But when I've talked to some of the staffers there, they're like, well, if one person says something, we're like, meh. If 10 people say it, they kind of listen. But if hundreds of people say something about the same thing, the staffers have no option but to bring it forward to their representative or their senator. So it does matter. Numbers matter. We need all the people we can get to speak up.
Yeah, if you hear some of the things he's said and done over the years, you'll be horrified. You don't want him. So HHS is in charge of Americans' health, basically. Right. You don't want him in charge of your health.
No.
All right, today we're going to talk about your immune health. And I guess there's a theme. It's like the immune system gone awry. Things are breaking.
Yes.
And we're going to start with a snippet from Cindy.
Yeah. So I picked a paper. I was trying to look for things that we don't talk about that often and that Brianne would be interested in. We were just chatting about the fact that Brianne is mystified by the fact that she feels she has allergies and is testing negative for allergies, and we can attack that on a different episode. But the paper I picked is called Interleukin-33 Activated Basophils Promote Asthma by Regulating Th2 Cell Entry into Lung Tissue. And it's got multiple authors that I can't really pronounce. So there's 2 equal contributors, Schultz and Gomez, and then the senior author is Bart Lambert. And I picked this paper because it's about asthma and allergy, and that's not a topic that we cover that often on Immune, and I think it's a really fascinating topic. Asthma is one of the most common lung diseases. About 25 million Americans have asthma. That's a lot of people. It's like 7-8% of the country has asthma. And it's a very unpleasant disease. For those of you who have it, you know this already. It's chronic. There's no cure. It's caused by inflammation in the lungs. And it's not the typical type of inflammation we think about when we think about inflammation, like to a bacterium infection or to a scratch or to something like happening in your skin. It's a different kind of inflammation, and we'll get to that. But this inflammation ends up with tightening of the muscles around the airways. So you get what's called bronchoconstriction, makes it really hard. Your airways have to open and close to bring oxygen in and oxygenate the blood. So it's really hard to breathe. There's a lot of coughing, wheezing, and shortness of breath. There's a lot of different associations, including genetic associations, so it's familial, but also lifestyle and environment. So pollution, smoking, dust mites, molds, occupational hazards, things like that can bring this on. But once you have it, you can't really cure yourself of it, but there are treatments. So if you have an acute attack of asthma, that's often what you'll think of. People will be like, they can't breathe, right? So they can take bronchodilators to try and open up those airways. But the way to control this chronic infection really is just kind of the sledgehammer approach to shut down the immune response. So steroid inhalers, or if that doesn't work well enough, oral steroids. So this
would it be like an EpiPen, right?
Not an EpiPen for asthma. So asthma it is somewhat also a type 1 hypersensitivity. So there's 4 types of hypersensitivities that we can talk about if you're interested. But type 1s are the IgE mediated. And this paper will actually address the IgE component because it was thought that there was an IgE component. But they're going to show that this is actually IgE independent. But anaphylaxis, so when somebody gets a bee sting or they eat peanuts or something like that, that is absolutely IgE mediated. And when that happens, that's when you give an EpiPen. And the primary reason you do that, as far as I understand, is because there's a blood pressure drop, because mast cells all over your body secrete histamine and other factors that cause massive vasodilation and blood leakage. And if you don't keep the blood pressure up, you will die because you can't oxygenate your tissues. So that's the main reason there. But this is a little bit different. This is more of just opening the airways. It's not the same kind of throat constriction that you would get if you eat a peanut. There's swelling of the throat. You need to open that up, and that helps with that.
So you would carry an inhaler if you have.
So you'd carry an inhaler if you had asthma, right? Okay. So if you have an asthma attack and you're having trouble breathing, you do an inhaler, and it causes the airways in your lungs to open up.
Are there any kinds of asthma that are not triggered by allergens that just happen?
Yes.
Exercise
induced.
So some people, when they exercise, then they have they get an exercise induced asthma and have trouble breathing and their lungs constrict up. Do you know of other ones?
That's the main one that I was thinking of.
Yeah.
I also thank you, Cindy, for talking about all of this, as I am lecturing on it later this week, and so it's a nice refresher.
It's always fun to review what I went back and I looked again. I'm like, oh yeah, that's interesting. I'm not trying to give you ways to treat asthma. If you have it, you obviously have to talk to your doctor. But the 2 main ways you do it are if you have an acute attack, you do this inhaler. That's basically just opens up the airways. But your longer term treatment is going to be some sort of steroid. So that's often in the form of an inhaler as well. So that's the daily maintenance inhaler of steroid. But I mean, you're putting a strong immunosuppressant into the lung, which is a major source of continuous exposure to potential pathogens. So it's not great. It would be better if we understood newer different ways to try and treat the disease that are much more specifically targeting the things that are driving the disease rather than just saying, let's immunosuppress, full stop, because that's not a great approach. But that's basically what we have right now. So the type of inflammation that's induced is different. It's called type 2 inflammation. So type 1 inflammation is what we think about when you have a microbial infection, like a bacterial infection or something like that. And that's associated with a lot of inflammatory cytokines, some of which are like TNF, IL-1, IL-6, but also interferon gamma that's produced by Th1 or T helper 1 cells, which is why we call this a type 1 inflammation. Instead, this is a type 2 inflammation. So type 2 inflammation, is also inflammation, but it's driven by different cells and has different cytokines and different outcomes. So the typical inflammation with type 1, you think of the redness, heat, swelling, pain, right? For the type 2 inflammation, it's a little bit different. It's goblet cells get involved, so there's a lot of mucus production. There's this muscle response, so this constriction of muscle. If it happens in the gut, it can cause peristalsis and cause things to move faster in the gut, so you can get diarrhea and things because you get fluids and mucus. If it's in the lungs, you get a lot of mucus and a lot of coughing and sneezing and release of all of these things. So they're mediated by different cytokines and cells. So the major cells associated with type 2 inflammation are eosinophils and basophils, and also mast cells, but we're going to focus on eosinophils and basophils. And they produce cytokines called IL-4, IL-5, and IL-13 are the main ones, and they will drive goblet hyperplasia, so more goblet cells. The goblet cells produce mucus. It also is involved in the vasoconstriction. It can also prime blood vessels for exit of cells, and it also promotes the production of IgE. Right, so we just mentioned that with anaphylaxis is IgE mediated, but this can also, type 2 inflammation can also be associated with IgE. So in the case of asthma, it's typically the finger is pointed towards eosinophils. They're the ones that go in, and those are the ones that are primarily accused of the hyperreactivity that's in the airways, the remodeling that happens, because if you have acute asthma attack after acute asthma attack, the remodeling that happens makes more fibrotic tissue that doesn't expand and contract as easily. So overall, your lung function decreases over time if you continue to have these acute attacks. And there's just a lot of mucus. So they primarily point the finger at these eosinophils. But really understanding exactly how all of this works is going to help us develop new therapies. And some things that people have tried, I mentioned, there's IL-4, IL-5, and IL-13. So people have tried blocking IL-5. They've tried blocking IL-13. And one of the things that's more recently working is blocking IL-4 and IL-13. And this is a drug that you often see on the internet. It's not for asthma necessarily, but it's for psoriasis and other things called Dupixent or dupilumab. And that's a monoclonal antibody therapy that targets IL-4 and IL-13. So it's one of the drugs that we have to target type 2 inflammation.
So this is infused, I guess, right?
I believe so, yeah. I think it's a...
So you would get periodic infusions to prevent...
Yeah.
Right.
Yeah.
Right. So asthma is not the only type 2 inflammatory disease. Psoriasis is another one. And I think Dupixent is really for psoriasis. If I remember correctly, I should have looked that up. So I can get corrected by our listeners. Okay. So the other interesting thing to connect to this is that IL-33, so this is a different cytokine. That's not those typical ones that you think of. Polymorphisms are associated with asthma. And so that's kind of interesting. Or the receptor for it, which very confusingly is called IL-1 RL1, so receptor L1, which is the receptor for IL-33, whatever. But that's what it is. But interestingly, IL-33 promotes Th2 cytokines, mast cell activation, and basophil activation. So if we think that these type 2 cytokines are important for driving asthma, and IL-33, there's a connection with asthma, and IL-33 promotes the production of these pro-inflammatory cytokines, then it might be kind of interesting to think about what is the role of IL-33 in this. So that's somewhat of what this paper does, is start to look at that. So the other thing they look at is basophils, because we kind of ignore them. I don't know how much you teach about them in your class, Brienne, but I usually just kind of ignore them.
I talk about them on the sort of cells of the immune system, kind of beginning of the semester. And I usually, we look at their numbers, and we say, look, there's so few of them in the blood. So there's not much we can say about them. The end.
Right. So it turns out that IL-33 can act on basophils. And basophils are, as you just said, very kind of interesting but understudied. They are definitely found in the asthma patient lungs. So if they take samples of patient lungs, they do not do that while they're alive, but they do that after, in a post-mortem. You can actually find these basophils that are in relatively high numbers in an asthma patient. We know from other studies that they can present antigen, which is interesting. And they do have Fc receptors to IgEs, so they can degranulate and contribute to this hypersensitivity type 1 and the anaphylaxis that I mentioned at the beginning. So we know a little bit about what they can do, but we don't really know what they're doing in asthma. So what this paper set out to address was the role of basophils in asthma, and they used what's called a house dust mite antigen model. And what they really wanted to know was, are there basophils there in this model? If they are, what are they doing? And if they're doing something, are they there and important during the initiation phase, or what we call sensitization, or the effector phase? And this is actually important, because if they're involved in initiating the response, there's not much you can do, because you don't know, tomorrow I'm going to breathe some and it's going to induce asthma in me for the very first time. We don't know that. But if somebody has asthma and we can know what cells are driving the pathological and acute response, we could think about blocking that. So if they have a role in the effector phase of it, we could think of targeting them. And the bottom line of this paper was that that's actually what they found. They found that if they depleted the basophils during the induction of a response, the basophils basically played no role. But if they depleted them during the sensitization phase, they could basically shut everything down. So the way they did this was they used what's called the house dust mite model. So house dust mites are the little microscopic things that live all over your skin and get deposited all over your body, and you vacuum them up as dust.
But they also consume some of the other stuff that's around that is the dust.
They eat your little dead skin cells, right? So they're not all bad. But
so they're in our eyelashes, our eyebrows and stuff, right? Oh my gosh. Yeah. It's weird.
They're pretty cool looking. They kind of look like really big, giant, I don't know.
Yeah, just Google it. You can
monsters or whatever. Yeah, they're cute if you think that kind of thing is cute. But anyway, so they developed this house dust mite model, and the way they do this is they take mice and they intratracheally instill in PBS or house dust mite antigen. Then they wait 6 days, and then on day 6, 7, 8, 9, 10, they challenge them intranasally with the house dust mite antigen, and then they analyze on day 14. So this is a well-established model. And what they were able to do was show that there was increase in eosinophils, lymphocytes, basophils in the lungs, IL-5 and IL-13 were increased. There was an increase in mucus. There was pathology associated with it. So all of these things kind of fit basically with your standard asthma. And they, I don't want to go into too much detail about all of the things that they did in this paper, because this is a snippet, but it was a really interesting paper. But what was cool was they used tools in which they could totally knock out basophils, or they could selectively knock them out at specific points in time. And they used a model where you can use an inducible Cree to knock out, put a diphtheria toxin and then knock out those cells in a specific time point. So if you inject in a drug, they then deplete their basophils. So then they could ask, well, what role did the basophils play? And they knocked them out when they were initiating the response. So during the initial intratracheal injection. And it didn't really have any effect. You could still induce the disease exactly the same. But if they waited and they depleted them during the effector phase, so when they did the exposure in the nose, now they could reduce every single measure that they had, including the lymphocyte and eosinophil recruitment, the mucus, the pathology. All of these different parameters were all reduced if they depleted the basophils during the sensitization phase. And then they did a whole bunch of other studies. But the key one was they figured that the basophils must be responding to IgE because they saw IgE in the blood and in the tissues. And they were like, oh, well, that must be mediating it. But they made a basophil specific knockout of the Fc epsilon receptor that actually had no effect. And they were like, well, if it's not that, then what is it? Then they went back and looked at IL-33, and what they were able to show is that they could use a decoy receptor that would bind up all the IL-33, and that reduced the recruitment of eosinophils and lymphocytes and IL-5 and IL-13. So it suggests that the basophils, which express the receptor, are somehow responding to the IL-33 and then driving the disease outcome. And the next thing they did was knock out IL-4 in the basophils. And they could say then that the basophils via the IL-33 response were then making IL-4. And that then promoted the recruitment of Th2 T cells that would produce the IL-5 and IL-13 and drive the recruitment of additional effectors and cause the actual disease. So the last thing they did was sort of go on and look at endothelium and see whether there was something about the ability to recruit cells, and there was a role for some integrins in this process. But basically what they did in the end was put together a model. And the model is that upon house dust mite exposure, there's IL-33 production that acts on the receptor, which is this IL-1, RL-1, or also called ST2. That's on basophils. So the IL-33 acts on the basophils. The basophils produce IL-4, and that acts on endothelial cells to help recruit Th2 cells. Then the Th2 cells come in and they start dumping out all the type 2 cytokines, IL-4, IL-5, and IL-13. And these are the ones that mediate the hypersensitivity response. So the asthma, this airway constriction, mucus hyperproduction, and the tissue remodeling that ends in the long-term sequelae of asthma. Now, the thing that this paper doesn't address is where does the IL-33 come from in the first place? I guess that'll be their next paper that they're going to work on. In mice, we know it can be produced by endothelial cells, fibroblasts, and other stromal cells. I'm just wondering whether it's maybe the epithelial cells in the lung that when they're exposed to the house dust mite, somehow they're making it and initiating this whole response. But then, you all come back to a chicken and egg, because then what in the house dust mite is driving this whole process in certain individuals and not other individuals, because not everybody gets asthma, but everybody gets exposed to house dust mite. Mm-hmm. So it leaves a really interesting set of questions. But I thought that this whole paper and using this model and then using the basophils knocking out at different times and kind of analyzing the molecular pathways of how all of this worked was kind of interesting.
Yeah, I also thought it was really interesting that the way that they tied in the epithelial cells onto all of this, because when I think about sort of IL-33 and IL-4 production, I say, oh, well, that's going to push a Th2 response.
Right.
And so the idea that that was going to influence the local environment, which was going to then act on the Th2s, was sort of like an additional step that I don't usually think about, that I think is an important one for us to think of, because we, as everyone starts to get more and more into immunology and tissues, thinking about all of those cell types is really important.
And they only looked at T cells, really, but there's lots of other cells in those tissues. What there's not a lot of in the tissues, though, are the eosinophils during the steady state, or even the basophils, right? Because they're really induced pretty dramatically in response to the model. So it's something about the exposure to the house dust mite antigens drives this initial recruitment of those cells that are then primed to precipitate this response over and over.
Cindy, do you think that this is allergen specific, or this would apply to other allergens too?
That's a good question. I would think it would apply to other allergens that tend to drive asthma. This one in particular is just a well-established model that they have worked out exactly the timing and the dosage and the route of exposure that will drive this hypersensitivity reaction in the lung.
Yeah, I think this is like back in the recesses of my mind, and I could not give you great citations on it or anything. But I feel like I remember reading that often with the house dust mites, some of the proteases, I think there's a protease like DERP2
that's
exactly.
Involved. Yeah.
And I think there may be some similarities between some of those proteases and proteases involved in other allergens.
There could be. I think I remember reading something at some point about proteases damaging the tissue, and that also maybe incites an initial inflammatory response that allows the development of immunity against those host dust mites, where just breathing in an antigen on a day-to-day basis should not drive an immune response against it necessarily. Usually we're going to generate an immune response to something that's dangerous. So I think that the mites have this protease, like you said, that will get secreted out into the lung somehow and then cause some little bit of damage. And then if they have similarity to proteases from other organisms, then presumably you would potentially have something cross-reactive that would also drive it to other microbes or other larger organisms that you could breathe in.
So they say at the end, this has implications for basophil-directed treatments.
Yep.
So what would that be?
Well, I think that the basophils are making IL-4. So definitely therapies that target production of IL-4 and sop it up or prevent the signaling might help in an instance like this, because it does seem that if they knocked out IL-4 in the basophils, they could interrupt the cycle. So it's definitely the secretion products that the basophils are making. Obviously, targeting things that the Th2 cells are making could also have an effect. Yeah. So things like the Dupixent that targets both IL-4 and IL-13 might be something reasonable to test. I didn't go look and see whether those are in trials for asthma or just for psoriasis.
Yeah. They say during asthma exacerbation. So it sounded like it would be an inhaler-based thing again, right?
Potentially, yeah. Or I guess if you take it on a regular basis, maybe it would prevent the flare, like the asthma attacks.
Yeah.
That it wouldn't necessarily, you wouldn't need a rescue therapy. So it's the idea of the rescue therapy that isn't very helpful, because once you initiate that inflammation, even if you stop it, you still have to repair the damage that's done. And that repair always ends up in remodeling the tissue, which then over time compromises the breathing. So if you can prevent having an asthma attack, it's always better than trying to rescue yourself once you have one.
Right.
I think also thinking about some of that basophil targeted treatment requires us to know more things about basophils.
That is true. So we should study them more.
Well, the first step is telling students about them so they get excited.
Yeah,
right? Exactly.
Thank you, Cindy.
Yeah.
What is it with the immune system? It's like delicately balanced and a little trip.
Yeah. I don't know how we're alive. Whenever I teach the veterinary students, they're like, how are we even alive? I'm like, yeah, that's a very good question. Yeah. I cannot answer that.
On one of the first days of all of my immunology classes, I tell my students the most important thing to really understand to be an immunologist is Goldilocks and the Three Bears.
Yeah.
That's
about
true.
Too much is bad. Too little is bad. It always has to be just right.
Yep.
Amazing. Yeah, it's cool. All right. Brianne's going to tell us about another problematic immune response.
I am going to tell us about another problematic immune response. And in fact, the authors address this by doing some of those conditional knockouts of things at specific times and in specific cells, just like Cindy talked about. So there's another way that these papers tie together. This is a paper from Cell called Neoself-antigens are the primary target for autoreactive T cells in human lupus. The first author is Mori, and the senior author is Arase. Most of these authors are from Osaka University in Japan at the amazingly named World Premier International Immunology Frontier Research Center. Though there are authors from some other universities in Japan, and from Riken and from a few other locations.
And since we're talking about defects, lupus is primarily a type 3 hypersensitivity reaction because and they're not really going to, I mean, they do touch on this because they do some studies looking at the anti-nuclear antibodies, but basically you get autoantibodies and it's immune complexes with those soluble autoantibodies. They form chains and clumps, and those clumps can get deposited and cause glomerulonephritis or cause like rashes in the face. And that's like the what lupus is.
Yeah. So in most lupus patients, there are these anti-self antibodies. They often bind to things like DNA or RNA or histones or a number of other kind of intracellular molecules, but that are also molecules that are quite prevalent throughout the body. And you can have these immune complexes of antigen antibody get deposited in a number of areas, just like Cindy mentioned. And because there are sort of differences in where the antigen complexes get caught up and exactly which antigens may be present in different patients, lupus often presents with somewhat different symptoms from patient to patient. And it's a disease that we really want to try to help patients with because it's not a very pleasant experience from my understanding.
Yeah. And it's often like diagnosed, you have to have a certain number of factors associated with the disease, but it's a lot of, well, it's not this and it's not this and it's not that. So it must be lupus.
Yeah. So in fact, it's sometimes referred to as the great imitator because it has symptoms in common with so many other things and can be hard to diagnose. And there is a somewhat funny compilation that you can find on YouTube from the show House MD, where the younger physicians are often decide that certain things are lupus.
Yeah.
And House MD always says, it's not lupus. And so there's actually a compilation of him yelling that over and over again.
It's not lupus.
That's out there.
So that one is sarcoidosis is another one, right? That they always say. Yeah.
In thinking about what's going on to cause lupus, one thing that has been observed is that there are specific MHC class 2 alleles that are important. And so that's part of it. But of course, everybody has DNA. Everybody has some of these self-antigens. And so why some people might make a response to these antigens while others do not is important. The authors also note that if you want to get an antibody response, like the antibody response that's seen in a lupus patient or any other antibody response, you also need to activate helper T cells because helper T cells are required in order to help B cells. And in particular, these authors note that they're particularly unclear about how the helper T cell might get activated in order to turn on autoantibody production in the case of lupus.
And it's important to say that, like if you're making DNA antibodies, T cells don't recognize DNA.
Right.
They only recognize peptides.
Exactly.
Well, they only recognize proteins or pieces of proteins.
Yes. So that brings up really well antigen presentation and MHC. And the authors also note that there have been some kind of ideas or previous evidence about the expression of this MHC presentation molecule called invariant chain, which they talk about as II. And invariant chain has two important functions. One of them is that it is basically biosynthesized with MHC class 2 and blocks the MHC class 2 peptide binding cleft, so that inappropriate antigens, particularly antigens from the wrong part of the cell, can't get onto MHC class 2. So the MHC class 2 binding cleft is covered by invariant chain until the MHC class 2 molecule gets to the right location in the cell for loading.
And this is because MHC class 1 will load peptides that it has in the ER, but we don't want to load up class 2 with peptides from the ER because we want class 2 to pick up things that get internalized.
Exactly. And that's a lot of how I think about it. I think that the authors were focusing more on, we also don't want self-ER resonant peptides getting on class 2. And that was something I had to think a little bit about that I don't think about as much.
Yeah.
And one piece that's sort of related to that is that MHC class 2 has open ends. So it can bind peptides of different lengths. In fact, in some undergrad immunology textbooks, when they talk about MHC class 2, they actually show a photograph of a hot dog in a hot dog bun.
Which is
Because
open-ended,
MHC class 1 is a little hot dog that's fully inside the bun. And MHC class 2 is like normal size hot dog bun with a 12-inch hot dog.
yeah, or an any length hot dog, because you can have variation.
I never saw a hot dog bun with closed ends. Yeah.
Oh, those lobster roll ones.
Really?
Yeah. So the ones where you get like lobster roll on it, they're kind of closed on the ends.
Oh, let's see.
I mean, you can break them open, but yeah.
And so the fact that, so they're kind of worried about maybe partially folded self proteins that are in the ER getting into that open-ended MHC class 2. And I hadn't thought about that as much before this paper, but that's something that the authors are thinking about quite a lot. Did you find the lobster rolls?
Yeah, some are open and some are closed. Yeah, depends. But I'm just getting hungry now looking at it. It's good. They look good. Better than hot dogs, right?
Yeah. So the authors note that previously people have made mice that are deficient in invariant chain. And so they never have invariant chain ever in their life. And those mice don't seem to have lupus-like symptoms, but they also have this idea that there might be something going on with invariant chain and lupus. So here they make a different mouse model where the invariant chain can be deleted when the mice are given the drug tamoxifen. So the mice are able to get to adulthood with invariant chain, but then they delete invariant chain from their cells later on in their life. And they spend some time talking about how complete the depletion is. And I spent some time thinking about that as well. But the moral of the story is, which they sort of show in one of the first figures, is that when they do this inducible invariant chain knockout, they lose invariant chain on some B cells, but not all of them. And they also get sort of reduced levels of MHC class 2, but it's not like a complete knockout.
So I wondered about this because I said, okay, this is cool. You can do this, but does it have any biological relevance? I'm like, why would you think about knocking invariant chain out later? But they come up with some good explanations to why this might be biologically relevant, right?
Yeah, they do. I was going to come back to that part at the end, but yeah, they totally come up with some reasons why your invariant chain levels might change in your life.
Right, which is important because just because you can do something doesn't mean it means anything.
Exactly.
Sense, right? But there is, put a pin in that. We'll come back to it. There is a biologically relevant, and they do study it. So
yes, exactly. And they notice that their inducible invariant chain knockout mice have bigger spleens, and some of them die. So it seems like there is sort of a different phenotype here than in the constitutive invariant chain knockout mice. Further, when they look at those invariant chain inducible knockout mice, they see that they have more germinal center B cells, plasma cells, activated T cells, T follicular helper cells. So it basically looks like they have a more sort of activated, perhaps hyperactivated immune response. They also seem to have production of a lot of anti-nuclear antibodies, which the constitutive invariant chain knockout mice do not have. And those include antibodies against single-stranded DNA, ribonuclear proteins, and histones. The inducible knockout mice also have complexes of IgG with complement proteins C3 and C1q. And so in the end, these inducible invariant chain knockouts where invariant chain was sort of removed in the adult look like lupus patients. human lupus patients, while the constitutive or whole life knockout of the invariant chain mice do not. So there seems to be something different when invariant chain was knocked out in the adult mice.
Right. So what is your take on why knocking it out all the time would have no effect?
I was super curious about that as well. So they kind of make a couple arguments about that in the paper. One of the arguments that they make that I think caught me the most was the idea that if invariant chain is completely knocked out, that means it's also completely knocked out in the thymus.
Yeah.
And so you are both impacting T cell development and maybe inducing fewer T cells to come out of the thymus that could cause problems in the first place. And also that you may, if some of this weird antigen presentation that we'll see is going on, it's likely going on in the thymus. And so you are actually tolerizing cells to these antigens where you didn't tolerize before.
Right. The key difference between inducing deletion later and its complete loss from the beginning is the fact that you have that education that's happening of the T cells.
Exactly.
Yeah.
And so did the T cells get educated to think these were okay peptides, or are they just now these new peptides that the T cells were not educated against?
Right.
So it looks like sort of inducible or a loss of invariant chain in adult mice leads to this lupus kind of phenotype. And their idea is that this is happening because perhaps cells in these mice are presenting self-antigens that they should not be presenting because they don't have invariant chain blocking the MHC class II cleft.
So it's picking up peptides in the ER where they shouldn't.
Exactly. It's picking up peptides in the ER where it shouldn't. And in particular, they're thinking it's picking up maybe some of these self-ER resonant proteins, maybe even that are, say, partially unfolded. They get stuck in the MHC and the folded parts are outside the cleft, outside the hot dog bun. So they go have their wild type mice. They have the constitutive knockouts. They have the inducible knockouts, and they have heterozygous mice that have some knockout and some regular invariant chain production. And they look at what kind of antigens are actually presented by MHC class II proteins in these situations. The big picture, I spent a lot of time on this figure.
Yeah.
For some reason, this figure really, really got me for reasons I don't really know. But when they have either the heterozygous or the constitutive knockouts, they have a reduced number of peptides being presented. However, when you actually look at that small number of peptides that are presented to see what the peptides are by mass spec, you find that they are largely self-peptides.
Right.
And you see that there are way more self-peptides being presented in these situations.
So a naive non-immunologist question. I thought all the self-reactive MHCs were gone, in thymus development. Right?
Well, so the idea is that the self-reactive T cells that were strongly self-reactive get deleted in the thymus.
Uh-huh.
And so, but the self-peptides may still get presented.
Okay.
Here we're seeing that maybe there are more self-peptides being presented or different self-peptides being presented in these mice compared to in a wild-type mouse.
Mm-hmm.
And so you're right. There's still the question of, do the T cells just deal with it? Or is this going to be a problem because we have new self-antigens being presented?
That you were not selected against because the ones you were, for class 2, the ones you were selected against for self-peptides were ones that were in endosomes or phagosomes or the M2C compartment, right? So you weren't selected against peptides that were in the ER that could bind to the MHC because the invariant chain blocked that.
Got it.
So during selection, you only got selected off the peptides that were in the M2C compartment, which is the way it normally should work. But then if you take away the invariant chain and the MHC can bind peptides in the ER, it's like they're they're new. They're surprising.
Right.
They look foreign, even though they're not, because you never selected against them.
So does this imply that lupus may in part be an invariant chain disease?
Yes.
That is part of what they're trying to argue here.
Okay.
Yes, that lupus might be a reduction in invariant chain disease, where these antigens that you hadn't seen before, they call them the neoself-antigens. That Cindy described really well as sort of the things you haven't seen before, surprise, because they're being loaded incorrectly, are being seen. And what's really important in figure 1D is they look at their heterozygous knockouts.
Mm.
And so
1D, yeah.
It was 2D, you're right, 2D. So these mice have less invariant chain, but not absolutely none.
Right.
And even in the mice that have less invariant chain, they start to see an increase in self-peptide presentation. So even if you have, again, less invariant chain, that's sort of enough to start presenting these neoself-antigens.
And it's not a lot, because the other key thing is that there's many fewer peptides.
Yes.
Overall. Because if you look at figure 2B, where they're showing you the wild type levels have like 300 peptides, whatever. These are the different the breadth of peptides, right? There's far fewer on the heterozygote and even less on the constitutive. Yeah. So there's just generally way less antigen presentation, because we teach you need invariant chain to stabilize MHC class 2 to get it out of the ER. Mm-hmm. Right. So it doesn't get out of the ER very effectively unless it has a peptide bound. And the peptide that's supposed to be bound is the piece of invariant chain. Right. So it's very inefficient antigen presentation without invariant chain, but it's not nothing.
Right.
And what they're arguing is that even though there's very little that gets out, the stuff that gets out is actually binding those peptides that are bad. There's these neoantigens, right?
Yeah, exactly. So now they want to look at sort of B cell responses, and look at the B cell activation in these invariant chain inducible knockout mice. They do some single cell sequencing, and they see that these mice who have inducible invariant chain deletion have some changes in their B cell number. They have fewer B cells, but they have way more plasma cells. So it looks like they have more activation, again, of their immune response. And when they look at specific transcripts, they find that the B cells in these mice have increases in transcripts that are very similar to what is seen in lupus patients.
So I had a little bit of a beef with this one. Because the genes that they picked are things like NF-kappa B. That's not a lupus specific gene. I've looked at, the gene signatures for lupus, and it's almost all interferon genes is what typically comes up. Now, I don't know what's specific in a B cell. It might be a little bit different, but I feel like C-Jun, CD69, NF-kappa B, FOS, these are just general inflammatory genes. They're not really lupus genes.
Yeah. The only thing that they mention about that, and this was one of those things where I don't know enough about gene signatures in lupus to know how much I believe or not, is that they say that yes, you can actually get many of these genes in other situations, but you can't get the STAT1. And so they try to hang a lot on the STAT1 difference in addition to the other genes. Again, I don't know enough about the gene signatures to know how much weight to put on that.
I don't know. That's the least upregulated to me if I look at the violin plots that they have in 3C. STAT1 is not the biggest difference. But I guess, you don't need a huge difference if it has a huge impact.
Yeah.
So I don't know.
No, I totally agree that there could be a lot more here. But I am not a lupus expert.
No.
So then they actually clone out some B cell receptors to look at the actual antibodies that are made in these mice.
And this is standard now too. Like I'm amazed every time I see this. I'm like, wow, that is not easy. No. But it's standard practice now. Like if you find changes in B cells, you go and clone the B cell receptors. If you find differences in T cells, you clone all the T cell receptors.
Yeah, and they do both in this paper.
And you express them, and then you test their function.
Right.
It's like, wow, that's really cool.
Yeah. So they tested the B cell receptors, and they first just looked to see if there was sort of clonal expansion of individual B cells in the mice. And they saw that the inducible invariant chain knockouts had more clonal expansion. So it seemed as though these mice were having their B cells make a response to something, even though they were not infected with pathogens.
Right.
It also seemed they also saw an increase in IgG class switching in these mice. So it looked like, again, these B cells had been activated, were starting to make antibody. And then when they actually, as Cindy said, made the antibodies and looked at their function, they saw that the inducible invariant chain knockout mice were making anti-nuclear antibodies, while the wild types were not. And so, again, sort of all of this together said to them that the B cells are different in these mice that had adult onset invariant chain loss. And it looked somewhat like lupus.
So they basically looked at antigens that bound these antibodies based on what you would find in lupus, right?
Well, so in lupus, one of the things that you do is you take a particular cell line and put it on a slide.
I think it's Hep
G2.
G2.
Hep G2. Mm-hmm. And then you add antibodies and you look to see if the antibodies bind to the nucleus.
Okay.
Are there nuclear? And so it's just sort of a very broad, do you have antibodies against the nucleus or not?
Well, they can actually look at, people get really good at looking at the staining pattern, whether it labels the nucleus or both the nucleus and cytoplasm or the cytoplasm, and they can kind of tell you, is it anti-DNA antibodies, anti-RNA antibodies, nucleoprotein antibodies?
Yeah, exactly.
It's pretty cool.
Yeah.
And
so
I think it's still a relatively standard diagnostic test.
It is absolutely a standard diagnostic test. Even now is just, do you have antibodies that bind to the nucleus?
To Hep G2.
Yeah.
Yeah.
So now they wanted to look at T cells in these mice. So they look at...
Because they got class switched, which means you must have some sort of T cells. So what are the T cells doing? Right.
Exactly.
Yeah.
So they look at T cell... so this is, again, this gets a little tricky. So they look at T cells from wild type mice and their ability to be stimulated by antigen presenting cells from the different other mice. And it seems as though the invariant chain inducible knockouts can stimulate T cells better. So they are able to activate T cells while wild type cells cannot activate T cells. And when they look more specifically at some of the T cells that are in either the wild type or the inducible knockout mice, they find that the inducible knockout T cells have an expansion of T follicular helper cells. And they also are more clonally expanded. So they seem to have evidence that they have undergone some expansion. So now they actually take some of these T cells and clone out the T cell receptor. So they're looking at T cell receptors from different groups of mice. And they put those T cells into another mouse, those T cell receptors into another mouse, and just look to see, are those T cells activated by using this GFP reporter. And the idea is they're putting them into a mouse that is not infected with anything. In theory, you put the T cells into this other mouse, and those T cells shouldn't be activated because they should be, as we've sort of talked about, not responding to self-antigens. But when they put in the T cell receptors from these invariant chain knockout mice, they find that those T cells are more frequently activated against wild type. So we have this population of T cells in this invariant chain knockout mouse that can respond to self-antigens. And there is not a similar population in the wild type mice. And they also do that.
And that was a lot of clones too.
There were so many clones. So much work. So many clones. And it was a lot of those clones showed this activation.
Yeah. I was surprised at how many.
Yeah. So it's not a super rare thing. This is just some of the clones they picked, and many of the random ones they picked had this ability.
So if we translate that to us, we have a lot of rogue T cells walking around that could be autoreactive if our invariant chain goes down.
Yeah. And they also took those T cells, put them into mice, and then looked to see if those mice started making anti-nuclear antibodies. And if the mouse received T cells with the invariant chain knockout receptors, they ended up making more anti-nuclear antibodies, specifically more anti-single-strand DNA antibodies. So it's all kind of tying together. And they look further. And so now they sort of come to Cindy's question. Well, they come to a question that comes before, I guess, Cindy's question. But they say, all right, this was great, but it was all in mice. What about in human patients with lupus? Yep. Is this also something that we can see in human patients with lupus? So they isolate T cells from human lupus patients. They clone their T cell receptors, and they make T cells with these T cell receptors. And they see that these T cells are able to respond a bit more, particularly if you also get rid of invariant chain. So those T cells are maybe a little more responsive, but if you put those T cells in a situation where they are looking at an antigen presenting cell that is missing invariant chain, those T cells are very reactive. So it seems like the lupus patients have a bunch of T cells that are, as Cindy said, rogue T cells ready to go if invariant chain goes away.
And are already expanded. So it suggests that this had actually happened in those people, because otherwise, because those T cell clones that they're going to isolate are going to preferentially be expanded ones from the lupus patients, right?
Exactly.
It's just by frequency.
Yeah. So both this has happened before and could happen again to cause more problems. And they don't see any of this in T cells from healthy controls.
So they look at 3 lupus patients, and they're all the same, what they find, basically?
They're different TCR sequences, but they all have the same functional outcome.
Yeah, they have the same sort of big picture, what happens of having this activation of invariant chain deficient, but it's not necessarily the same antigen.
I just wonder if they looked at more. I mean, could there be heterogeneity in the mechanism? Because 3 is not a lot, right?
That's true.
No, that is true. Yeah. I guess they were kind of hoping that there would be like what we call a public clone, where lots of people have this. And if it got activated in the lupus patients, maybe you could see it much more frequently. Yep. But my understanding of it was that they didn't find a clone that was represented in more than one of those 3 people. That doesn't mean that if they screened a larger group of lupus patients, you might find a clone that's actually shared, or more than one clone that's shared.
So they did more work looking at the T cells in the lupus patients. And this was really getting at some of those activation questions that Cindy mentioned, to show that many of those T cells had been activated by invariant chain deficient presented antigens. And they also talk about the fact that perhaps there are more T follicular helper cells, there are more clonal cells. But I really want to mention the next part, because I think this is the part that both addresses Cindy's question, and I think that Vincent's going to like this part too.
Yeah.
So all of this is great. But again, we have this question of, well, okay, so this happens if you have a loss of invariant chain as you are an adult. And we can make that mouse through fun genetic engineering techniques. But does it actually ever happen where invariant chain production is reduced in a person? And they mentioned a little bit of this in their intro, but this figure is really where they sort of start to test it and look at what's going on. And they show that in individuals who are infected with Epstein-Barr virus, EBV, when that virus is in a lytic phase, so when it starts to reactivate, that reduces expression of invariant chain on those infected cells. And they, so they also talk about, what percentage of us have Epstein-Barr virus latently infecting us, and the fact that
Pretty
virus,
high, right?
Yeah, it's super high.
Over 95%. Yeah.
And that virus sometimes goes from latent infection to a lytic infection and reactivates. And
do we know what triggers that?
No,
stress?
not really. Stress, but we don't know the mechanism. It's really worked out for herpes simplex, but yeah, stress. That's it.
Yeah. They note that some people will have more frequent reactivations than others. And that there is epidemiological evidence that says people who have more frequent reactivations or lupus patients have more frequent reactivations. I was going to flip that over. But the lupus patients tended to have more frequent reactivations. So they say, well, maybe what's happening is when EBV reactivates, you're reducing your invariant chain levels and now starting to present these new antigens. And this is letting you make a autoantibody or autoimmune response to the neoself-antigen that you didn't make before. So they look at donors and their invariant chain levels, donor cells after EBV infection. They also look at cell lines with or without EBV and show that when you stimulate EBV positive cells to mimic reactivation, those cells lose invariant chain. If you look at EBV negative cells, then there is no change when those cells are activated. And they look at a gene that's involved in reactivation, BZLF1. And so when BZLF1 is being produced, when there's reactivation, there's a loss of invariant chain. But when you don't have that reactivation, there is no loss of invariant chain. They look at the peptides that are presented in the EBV reactivated versus the non-reactivated B cells. And in EBV reactivated B cells, you again start to have a lot more self-antigens being presented. And if you actually look at the responses of cells either to reactivated or non-reactivated EBV infected cells, you can see that there is a difference. And so they sort of have this idea that EBV reactivation is reducing invariant chain levels, and that is allowing for these self-reactive immune responses to happen.
I guess it's a form of immune evasion, right? To keep it down.
Exactly. That they talk about that a little bit in the discussion about how you could imagine how the virus would have evolved to perhaps reduce invariant chain because it doesn't want to get its peptides presented. And they show, at the beginning, it does really impact how many peptides are presented. So it's a mechanism of immune evasion, but it has sort of this side issue of inducing some neoself-reactive T cells.
It's just amazing. It's an amazing coincidence, right?
Yeah.
That because herpes viruses in general do a lot of downregulation of MHC and different mechanisms.
Right.
I think that's how we learned about most of antigen presentation, right? Because different viral proteins affect the different stages of antigen presentation. People could see that, and then they could isolate all the different viral proteins and then identify each of the different steps in antigen presentation.
Yeah, I have a slide in our textbook that has them all on, and I think invariant chain is one of them.
Yep.
keeping it in the ER. Oh, that's so cool. And then it's useful for understanding the lupus mechanism. That's very cool.
Yeah. So I just thought that was really interesting, both because I hadn't really thought about invariant chain loss leading to self-peptide presentation. I always think about it as
didn't think of that.
Yeah, I'd never thought of that.
Reduced antigen presentation.
Yeah.
Right? Just reduced. I didn't think about a switch in what peptides might be presented.
Exactly. I hadn't thought about that at all. I hadn't thought about the idea that EBV was going to influence invariant chain. I hadn't thought about how this could impact lupus. Like all of these cool pieces came together with this paper.
But not everyone that has an EBV reactivation develops lupus, right?
Nope.
Do you know the fraction? What is it roughly?
I think they did the other way around, right?
Yeah,
I don't know. I don't think they can know, like, if you get an EBV reactivation, how frequently you get lupus. But I think what they were able to say is that people who get lupus have more frequent reactivations than people who don't.
Right.
So it's not
So
exactly
how much
the
more?
answer.
Do you know?
I don't know.
I think
Because
they
that could tell
said
us
it.
if there's more than one mechanism, right?
Right. I think they mention it, but I don't remember where that was.
Interesting.
Yeah, I don't remember.
Because
But-
I know, also in COVID, some people get lupus, right? And I wonder if that's because of EBV reactivation or what. Something
Potentially.
else.
Could be.
There is some data about EBV reactivating during COVID.
Yeah.
What was I going to say? So part of it, Vincent, would have to do with, do you have the problematic MHC class 2 alleles that maybe, so you have to have the right MHC class 2 alleles. You have to have EBV reactivation, maybe at some frequency, along with other stuff that we don't know about.
Got it.
Yep.
Cool.
I learned a ton from thinking about this paper.
I really found this fascinating.
Yeah.
Then I know I asked you this question because I thought it was really interesting that they really didn't call them neoself-peptides.
Mm-hmm.
And it wasn't entirely clear to me what they think is binding to the MHC. Because you mentioned that the MHC is open on both sides for class 2.
Yeah.
And you kind of described it as a peptide across there and then maybe the folded part of the protein out the side.
Mm-hmm.
But it wasn't clear to me whether they were thinking that the TCR could actually bind some other three-dimensional structure than just a linear peptide in the MHC.
Yeah.
That wasn't entirely clear to me, and I'm not real sure.
Yeah. I mean, they sort of hint at this idea of a whole peptide getting presented in a few places, but none of their data really addresses that.
But they were so careful to call it like a neoantigen, but not really a novel peptide.
Well, they were also, I think, trying to be very careful about the way that we talk about neoantigens and tumor immunology, and talking about how this isn't the same kind of neoantigen that the tumor immunologists talk about.
Yeah, that's an important point because in tumors, there's mutations that happen or translocations of chromosomes and things where you get truly a new antigen.
A new protein that wasn't made before.
A new protein that wasn't made before. So you might get a novel peptide that is now recognized as foreign.
Right.
And that's different from this because these peptides would always be made, but it's just that they're not usually loaded onto the MHC2 because it's blocked until it gets to the M2C compartment.
Right.
So they make this statement in the discussion that infection with EBV or HCMV, which also downregulates in variant chain, does not induce autoimmune disease in most individuals. But what they do see is that viruses that establish persistent infection are more likely induced or reactivated in people with autoimmune disease than in healthy individuals. So they have more frequent reactivation. So they say maybe these neoself-antigens that are repeatedly presented in that fraction of patients are stimulating neoself-reactive T cells.
Exactly.
So that's very interesting. Yeah. Neat stuff.
Yeah.
All right. Thank you, Brianne.
Yeah. Thanks, Brianne. That was fun.
problem.
I enjoyed reading that paper.
How the immune system can harm you.
Yes.
Yep.
But we always
It's amazing
knew that.
we're all alive.
The immune system is a double-edged sword, right?
It's incredibly powerful, but it's incredibly dangerous and deadly too.
Yes.
Probably more so than many other systems, right?
Yeah, I think so.
I think so. I mean, I think that it is responding to something that's a really big evolutionary threat in microbes, especially microbes when we're young. So it has to be very, very powerful. But it also has to be really tightly regulated because of all the danger.
And things happen. Mutations happen. All right. That's Immune 87. You can find the show notes at microbe.tv/immune. If you have any questions or comments, immune@microbe.tv. And if you like these programs, consider supporting us, microbe.tv/contribute. Cindy Leifer's at Cornell University. Cynthia Leifer on Threads. Cindy Leifer on Blue Sky. Thank you, Cindy.
Thank you.
Brianne Barker's at Drew University. BioProf Barker on Blue Sky. Thanks, Brianne.
Thanks. I learned a lot.
I'm Vincent Racaniello. You can find me at microbe.tv. Music on Immune is by Tatami. Thanks for listening to Immune, the podcast that's infectious. We'll be back next month.